I'd want to welcome John Holloszy today to talk with us on the ACSM's Distinguished Leaders in Sports Medicine and Exercise Science series. And thanks for bearing all the heat and humidity to get over here, John.
Very happy to do it.
I just wanted to start out a little bit with your early life, really. Where you went to high school, the decision to go off to college, what you majored in, graduate education, that type of thing, if you were involved with sports or exercise at that time, and work our way up sort of chronologically to a point where your career and ACSM crossed paths somewhere along the way. So that would be great if you could take us through some of that, John.
I went to grade school and high school in India, where my father worked for the Maharaja of Bikaner. And I came to the States to go to college when I was 17.
You were born in India?
No, I was born in Austria, in Vienna.
Okay.
My name's Hungarian, but most Viennese are mixtures of Czech and Hungarian or Italian. You know, Vienna was the center of the empire, and everybody migrated.
Yeah.
So I'm a mixture. So I came over to the States by myself when I was 17. I started out as a aeronautical engineering student at Oregon State University, and decided that I didn't like engineering and switched over to pre-med.
And this is in Corvallis?
That's in Corvallis, yeah.
What year was that, John?
That was in 1950 that I started college.
Okay.
And I applied to medical school during my third year of college and was accepted by a number of medical schools, but decided to go to Washington University in St. Louis. I went through medical school there, graduated in 1957, and then took a internship and medical residency and training in endocrinology and metabolism, all at Washington University Medical Center.
Was there one or two people that you worked with there as
no, when you're a resident, you just during my third year of residency, I did do 6 months of research with Bill Dowday, who was head of the endocrinology department and was very well known in the area of growth hormone, IGF-1 signaling. And during my residency, I decided that I really didn't want to practice medicine. I was more interested in research and in preventive medicine. During my second year of residency, I applied to the National Institute of Aging for a postdoctoral fellowship, and was told that they would be very happy to have me there as a full-trained physician, because most of the people there were PhDs doing studies on humans. And I said, you understand, I'm not a U.S. citizen, and I know that you have to be a U.S. citizen to be a public health service officer, but I will be a citizen by the time that I come to work with you. They said, no problem. And I had to fill out a bunch of forms about my interests and so on.
What year was that, John?
That was in '59.
And where were they located, or where were you going to go?
I would have been in Baltimore, actually. That's where the Gerontology Research Center is.
Okay.
Well, right at the end of my third year of residency, they contacted me and said, we're sorry we made a mistake. You'll have to reapply again after you get your citizenship. So I stayed on for another year as chief resident. And early during that last year, I was contacted by the associate director of the Heart Disease Control Program, which at that time was a major component of the Public Health Service. It's a defunct now, but then it was a really big program. And this was right at the height of the coronary heart disease epidemic. Mm-hmm. And most of the public and most of the medical profession at that time thought that exercise was bad for you, that it accelerated aging, and you heard statements like, whenever I feel like exercising, I lie down,
yeah.
or the only exercise I get is carrying my active friends' coffins at their funerals, the sort of thing. Mm-hmm. And since I was interested in preventive medicine, and coronary heart disease was the #1 medical problem, I became interested in that, and I became convinced that physical inactivity was a major contributor to the high incidence of coronary heart disease. Somehow the form that I filled out when I applied to the National Institute on Aging was sent throughout the Public Health Service and came across his desk, and he saw that I had an interest in exercise and coronary heart disease. And he had the same interest.
Who is this?
This was a guy named Sam Fox.
Oh, I remember. Yeah, sure.
Remember Sam?
Yeah, he was highly associated with the college.
Exactly. Yeah. So Sam had been in touch with Tom Curitin, who ran the exercise physiology and fitness lab at the University of Illinois, and was one of the pioneers in getting people to try to exercise. Mm-hmm. And he and his graduate students were running an noon exercise program for faculty members and businessmen from the Corvallis, Corvallis area. Excuse me, not the Corvallis area. Champaign-Urbana area. Yeah. And he wanted to station me there to see if I could get some information on these participants. And with the help of some of his graduate students, including Jim Skinner, who I think is quite active in ACSM now, I was able to organize a couple of studies on most of the middle-aged men who were involved in the exercise program. And we got some very interesting results. We were the first to show that exercise lowers triglyceride levels, that a bout of exercise is followed by a decrease in triglyceride levels. And we also found, using a methodology that's not used anymore called ballistocardiography, that exercise improved cardiac function. And I presented let me backtrack. The thing that interested me most was the remarkable increase in exercise performance that occurred in these individuals. When they started out, they couldn't run a quarter of a mile. And after 3 months of training, they were running 4 miles at a pretty good clip.
Wow.
I thought, what the hell's going on?
Yeah.
And at that time, it was thought that the improvement in exercise performance that occurred with training was due entirely to an increase in cardiac output, with an increase in the ability to provide the working muscles with blood and oxygen. And it seemed obvious that that played the major role in the increase in maximal oxygen uptake that occurred with training. But I didn't see how it could explain the improvement in exercise capacity at the same submaximal intensity, say running 7 miles per hour, because cardiac output and blood flow to the muscles is the same in the trained and untrained state at the same submaximal work rate. Actually, slightly less in the trained state. So I thought there must be something going on in the skeletal muscles. Mm-hmm. And I had a fair amount of free time, so I spent quite a lot of time in the library trying to find out some information about adaptive response to training. There was really nothing. But I ran across a couple of papers. In one that had compared muscle from wild rabbits to domestic rabbits, and the other that compared muscle from wild ducks to domestic ducks. And there was a huge difference in mitochondrial content of the wild and domestic animals. And since they were the same species, I didn't see how this could be on a genetic basis. I thought it was probably due to huge difference in physical activity.
Yeah.
And so I came up with the hypothesis that exercise induces an increase in muscle mitochondria. And so at that point, my career took sort of a side path. I'd been concentrating on preventive medicine, you know, the health benefits of exercise. But I became fascinated with this possibility or question. And in order to study it, I realized I needed some training in biochemistry. So I made a visit back to St. Louis and talked to my old biochemistry professor, Calorie, who was towards the end of his career. He was a very famous biochemist. He was a Nobel Prize winner. He didn't remember me as a student, which is not too surprising. But he was very welcoming and said that they would be delighted to have me come work with them, but that there was nobody in the department working with mitochondria, and that I should talk to other people in the department, see if there was anything that was going on that interested me. And one of the assistant professors, a guy named Narahara, who had been Corey's postdoc, and I was still working with Corey as an assistant professor, was doing studies on frog sartorius muscles, a very thin muscle that had been used for years to study muscle contraction. And he was using it to study the effect of insulin on glucose transport. And since I was also interested in diabetes, I said, you know, this would be a very interesting model to study the effect of exercise on glucose transport, because there's some evidence that exercise lowers blood glucose levels. So I spent the next 2 years studying effective exercise on, or muscle contractions on glucose transport in epitrochlearis muscles.
When would that have been? I've lost track.
That
of
was
time.
between 61 and 63.
Okay, so when you were in Urbana, Champaign-Urbana.
Yeah, that was from 1960 to 1962.
Okay, and were you on that public health service grant then, when you were up there?
I was a public health service officer.
Okay.
And public health service officers are also physicians for the Coast Guard. So I was a lieutenant commander in the public health service.
Okay. And then did you leave that role then when you went to St. Louis?
I completed my tour of duty after 2 years.
Okay.
And then arranged to take a postdoctoral fellowship with Corey.
Okay, I'm sorry I interrupted.
No,
so you're back in St. Louis.
So 61 to 63, I was in the public health service. 63 to 65, I was a postdoc in
and this is the
biochemistry.
sartorius and what you were saying. Okay, thank you, John.
And that became another major area of interest, and I've been studying the effect of the insulin-like effect of exercise on muscle glucose transport. It's one of my interests ever since. While I was in the biochemistry department, the studies that I had done in Illinois received a huge amount of publicity because there was hardly any exercise research going on. And the finding that exercise lowers triglycerides and improves heart function created a lot of interest in the articles in Time magazine and Newsweek, and my mother collected a whole...
Oh, fantastic.
Anyways, the chairman of the preventive medicine department at WashU saw some of this information and remembered me as a medical student since I'd done some work in the preventive medicine department. And the major emphasis in the preventive medicine department was on nutrition. And he said he'd like to build a parallel track on the health benefits of exercise.
Oh, wow.
So he wanted me to start a exercise research program. So after I finished my postdoc, I set up my own lab in the preventive medicine department. And the goal was to build a facility, both to train people and to do human exercise research.
Mm-hmm.
And it took about 5 years to raise the money to build that facility. So during that time, I just concentrated on basic research. And the first study that I did involved having a treadmill built and learning how to run rats on a treadmill. That was the most difficult part of it. But we were able to, I had a high school student working with me, training the rats. We started them out running 10 minutes a day, and over a 3-month period, gradually increased the amount of exercise they were doing until they were running 2 hours at 30 meters per minute up a 12-degree incline, which is a hell of a lot of exercise for a rat.
Yeah.
So these were highly trained rats.
Yeah.
And during that time, I was also setting up the methodology for measuring mitochondrial enzymes and oxidative capacity in muscle. And when we killed the first 2 rats, and skinned the hind limb, it was obvious that something very major had happened, because there was a huge difference in color between the muscles. The muscles of trained animals were a much deeper red color than that of the untrained animals, which is exactly the same thing that had been found in the wild ducks and the or wild rabbits and the tame rabbits.
Mm-hmm.
So anyways, I found out that there was about a 2-fold increase in mitochondria and about 2-fold increase in ability to oxidize substrate, indicating that there was an increase in functional mitochondria. And I was able to publish that paper in the Journal of Biological Chemistry. And that, of course, aroused a lot of interest in the exercise physiology.
Was that in 1968?
That was 67.
Because I've talked to Ken Baldwin and Ron Turyoung, and everyone keeps referring to this paper in 1968. So
It was actually in 67.
Okay.
And that aroused a lot of interest in the exercise physiology field and attracted a lot of postdoctoral fellows to come work with me who had been trained in exercise physiology.
Yes.
And who actually knew more about muscle than I did at that time. And that, of course, speeded up progress, research. And I spent the next 3-4 years just concentrating on this adaptive response. And after we'd clearly demonstrated those increase in mitochondria, I was also able to get at the mechanism by which the increase in mitochondria results in an increase in exercise capacity. The mechanism by which it slows glycogen depletion and decreases lactate production. And the mechanism involves a smaller decrease in high-energy phosphates with a smaller increase in ADP and inorganic phosphate and AMP. And it's inorganic phosphate and AMP that regulate glycogen breakdown and glycolysis. There's also a huge increase in the capacity to oxidize fat. So what happens in the trained state, there's a carbohydrate sparing effect and there's increased oxidation of fat. And 2 of the factors that lead to fatigue are increased lactate production early during exercise and glycogen depletion late during exercise. Mm-hmm. And the effect of the increase in mitochondria is to decrease lactate production and slow glycogen.
So it offsets.
Yeah. It's about 1970, the human exercise facilities became available. And I returned to my original interest, which was exercise and coronary heart disease.
Was this still at St. Louis?
Yeah, my whole career has been at Washington University.
Okay.
I started as a medical student in '53, and I'm still there, so
Okay.
I've been there longer than anybody. So I want to test how it had been shown. There were some people already using exercise to rehabilitate coronary patients, and they were using relatively brief periods of low-intensity exercise. It had been found that exercise training raises the angina threshold so that you have to exercise at a higher intensity before developing ischemia. And the reason for that is that training results in a slower heart rate and lower blood pressure at a given exercise intensity. So the heart's doing less work after mild training at the same exercise intensity. But this was not due to an actual adaptive response in the heart, because if you exercise at intensity that resulted in the same heart rate and blood pressure that caused the ischemia prior to training, you still got the ischemic ST changes and the angina. And I thought that with more intense and prolonged training, it might be possible to get an increase in coronary diameter and also in collaterals. And I was able to recruit a cardiologist to come work with me. And we told the hospital that we'd be happy to run a coronary rehab program for the hospital, and we had a nice exercise facility. So we ran a coronary rehab program between 4 and 6 every day for patients who were paying the hospital. And from these patients, we recruited individuals who wanted to be in our studies. And the motivation was that they didn't have to pay. And with the help of some of the postdocs who are training in exercise physiology and knew how to train people, and with this cardiologist supervising what was going on, we were able to bring these coronary patients to the point where they were doing a lot of exercise, running as much as 4 miles a day. This was very gradual.
Yeah.
But they actually became master athletes. I mean, they were running 4 miles a day or as much as 20 miles per week. And we had them do this for a year and found that at the end of a year, they either were not getting ischemia during exercise or were getting ST depression at a much higher heart rate blood pressure product. So there's actually improvement in blood supply to the heart. In the second study, we were able to show that they no longer had exercise-induced wall motion abnormalities. What this means is that when an area of the heart becomes ischemic, is not getting enough oxygen and blood, it stops contracting normally, so that when the rest of the heart is moving inwards, this area of the heart is bulging out.
I see.
And that disappeared, also providing evidence that the ischemia was gone.
Yeah.
And we subsequently did studies on patients with renal failure. This was prior to dialysis, and it had a really remarkable effect on them. It increased their hemoglobin levels and lowered their BUN levels. And it looked as though this would be a really useful treatment of patients who had chronic renal failure. But it really never took off because renal dialysis was developed, and people would rather sit and get dialysis
Than exercise.
And of course, it's more effective than the exercise. We did studies on diabetic patients, a lot of work on prevention of frailty in older people. We started out doing a lot of studies, just exercise physiology studies on younger people, and then worked our way up gradually to middle-aged people, master athletes, and then to very old people. And most of these studies on old people were supported by the National Institute on Aging. because of the possibility that this might be a useful way of keeping people out of nursing homes, keeping them at home independent. And we showed that young old people, people in the 65 to 75 year age range, with an appropriate training program, were able to adapt to training very well with really remarkable improvements in performance. And then we moved on to old people in the 75 up range, and tried to recruit people who had mild degree of frailty, were already having difficulty with activities of daily living such as shopping, taking care of the home, and so forth. And you can grade the degree of frailty with a series of tests: how long it takes to climb a flight of stairs, how long it takes to put on a coat, standing up from a chair. And with a year of training, which involved first stretching and flexibility, and then weight training, and then we added some endurance training. After a year of training, a lot of these people who had initially been frail were no longer frail, so it was effective too.
John, I've got a couple questions. When you met Sam Fox, did you know Joe Wolfe and Bert Hyman and Louis Bishop? They were all cardiologists that were founders of the college.
No.
You didn't know? Okay. And when you had when did you have your first postdoctoral student?
In '68.
Okay. Were you able to get funding for those students over the years?
Yes.
And how did you do that? Through your grants?
I got a training grant from the Aging Institute.
Okay.
And the rationale was that we needed to train people in the exercise area to be able to do research that was applicable to prevention of disease and maintenance of function during aging.
Okay. And so
and actually it was through my students, my postdocs, that I became member of the ACSM.
I wondered about that.
But I didn't come from an exercise background.
Right.
And I came to exercise physiology from a different perspective, and they had come from a sports background.
Yes.
And through them, I became a member of the ACSM and became quite active in the ACSM because that was the major source of my postdoctoral fellows for a long time.
Yeah, so you had some from Tipton's lab.
Yes.
Some from Curitin's lab.
Yes.
And other places, I'm sure.
Yes.
I remember John Ivey was a doctoral student when I was at Maryland, and I think he worked with Dave Clark. And I think he ended. John worked at your lab.
Yeah, he worked with me for. And I got quite a lot of Dave Costel's.
Yeah. Okay.
Graduate students.
Okay. Yeah, and somebody the other day said that you had like 90-some postdocs over the years.
I've had 90 postdocs.
Wow. And do you have any right now? Do you have people working with you now?
Yeah. My problem right now is that I don't have a training grant anymore.
Okay.
And in order to support a postdoc, you've got to pay them $35,000. That's the rule.
I see. Okay.
And so recently I haven't had any American postdocs. I've had mostly Japanese and Korean postdocs because they bring their own
funding. Yeah.
And over the years, I've had 8 Japanese postdocs. And the early ones are now professors and big shots back in Japan. And they're sending me their students.
Oh, that's neat. That must be rewarding.
Oh, yeah, it's a lot of fun.
So you started coming to some ACSM meetings then?
Yeah, I started coming regularly and participating in symposia and so on. Yeah, and I won both the ACSM Honor Award and the
citation.
Yeah.
And I think I remember at one point you were editor of ESSR.
Yeah, for 10 years.
For 10, okay. No wonder I remember it.
That's when we still had the red
The
book.
big red book,
Yeah.
yeah. I still have a
I
line on my
have-
shelf.
me too.
That's good stuff.
Yeah.
That's really good stuff.
Yeah.
So you're still at your lab, and how things have changed over time? What are you looking at now?
I'm still doing some exercise research, but most of my research now is more aging oriented. And the Aging Institute has become interested in effective calorie restriction in humans because there are many, many studies in animals showing that calorie restriction slows aging. You want to go into that?
Sure, a
Yeah.
little bit. Yeah, that's very interesting. Because you would sort of think it would be just the opposite.
You would.
I would, yeah.
Yeah, if you feed a rat or a mouse about 30% less than they would normally eat, they end up living 20-30% longer than control animals.
I need to cut back on my dinner.
And it's not only average longevity, it's maximal longevity that's
Wow.
increased. So about 9 years ago, the Aging Institute put out a request for applications to do studies on calorie restriction in humans. And I just lost funding for my Pepper Center, so I needed a large grant to keep my human research going. So I applied for that and got funded. And 2 other centers also got funded, Tufts in Boston and the Pennington Center in Louisiana. And in the initial study, we designed our own experiments. And what I did was to compare 20% calorie restriction with a 20% increase in energy expenditure without an increase in calorie restriction.
Okay.
So I had 2 groups of middle-aged people, one doing diet and one doing exercise. And this was followed by a 3-center study which is being done in collaboration with the Aging Institute on 2 years of 25% calorie restriction in 22 to 50-year-old people. And that study is starting to wind down. It hasn't been much fun for me because we didn't have much input into the design of the study, and we're not getting any feedback. We just send the blood samples, and we won't know what happened.
You don't know what's going on. Yeah.
But what has been a lot more fun for me is that about 8 years ago, about the time that we were getting started on the first calorie restriction study, I was contacted by a gentleman who said that he was a member of the Calorie Restriction Society. And he said he'd been doing severe calorie restriction for a number of years and wanted my advice on what kind of exercise he should be doing to try to maintain some muscle mass. I didn't know there was such a thing as a Calorie Restriction Society. And through him, we contacted the Calorie Restriction Society and recruited members of the Calorie Restriction Society to come to our lab to be studied. And we didn't have any funding for this. But a lot of these people, all of them that we've been in contact with are highly educated. They're all college graduates. A lot of them are professionals of various types, wealthy businessmen. These are all people who have worked very hard to educate themselves both about nutrition and aging. And they all read a book by a guy named Roy Walford called The 120-Year Diet. This was a guy who had done research on mice showing that it increased longevity. And he was claiming the same thing would happen in humans.
Okay.
And they bought into that.
I see.
So we gradually built up a group of 40 of them, and they come to St. Louis at their own expense, either when they're on their way somewhere else or specifically to be tested. And on average, they've been on calorie restriction 7.5 years, some of them much longer. The shortest time is 3 years. And they're showing all the same adaptive responses as the calorie-restricted animals. Very low levels of inflammation, blood pressure like little kids, 100/60. No disease risk factors, very high HDL cholesterol, low LDL cholesterol. We did muscle biopsies on 15 of them and that have been done gene chip analyses on them. And it showed a huge downregulation of all the genes involved in inflammation and in the pathway leading from insulin and IGF-1 to protein synthesis and cell division. So, and it's this pathway that seems to be involved both in cancer development and in aging. So that research has been much more interesting than the NIH-funded research.
Have you published much in this yet?
Oh, yeah.
Yeah. What journals?
Aging, Cell, Endocrinology, Age, Experimental Gerontology, PNAS. My colleagues, Luigi Fontana, who is doing most of this work, just published a review article in New England Journal.
Okay. Well, sort of the final thing is you've been involved in the American College of Sports Medicine quite a while now,
Yes.
and I've seen a lot of postdocs and their students. And where do you think the college is, and where they're going, and how do you still see it as an important organization and a vibrant organization? And
You have a few more minutes?
yes, sure. Yeah.
When I started out doing exercise research, my older friends and mentors in research said, for God's sake, don't say you're studying exercise. You're just using exercise as a tool to study mitochondrial biogenesis and regulation of glucose transport. This will ruin your career. Mm-hmm. So I said, the hell with that. My goal is to make exercise a reputable area of research that can get funding from the NIH, because it's an extremely important area, and it needs to be recognized as a legitimate area of research. Mm-hmm. So I stopped publishing in JBC and other journals, started publishing in Journal of Applied Physiology, and my postdocs, when they went out, they were able to get NIH funding. And I think that their involvement in ACSM and their students have played a major role in making exercise a legitimate, respected area of research. And I think ACSM has played a major role in that.
Yes.
Yeah.
Yeah. I agree. That's a very good point.
Yeah. So my association with the ACSM has been part of my original desire to make exercise a reputable area of research, which it was not.
Right. Absolutely.
Yeah.
Yeah.
People thought it was just sports, you know.
Just playing
Yeah,
games, and no significance to
Yeah.
it.
Yeah.
Well, time went really fast. I think we.
It has for me, you know, the last
Well, thank you so
50
much.
years have gone by very quickly.
Yeah. Oh, thank you, John.
You're very welcome.